I was only there for like 4 days.
All right. All right. So, welcome everybody. I'm very excited to have with us today the Lambert High School iGEM team. So, we're going to start off with them introducing themselves and giving their talk, and then we'll open up the Q&A for anyone who would like to ask the team questions live. So, take it away, everyone. You guys are muted. Um, you're still muted.
Hi. Hello.
There you
There
go.
you go.
Perfect. Okay, we can hear you. Go ahead. Thank you.
Okay, ready to start?
Why don't you introduce yourself?
Yeah. Okay, so I'm Rohan.
I'm Shashwat.
I'm Kirtana.
And I'm Veda.
And who are you?
Oh, we're part of the Lambert iGEM team here. And we were the ones that presented the live stage presentation at a Jamboree. So
We'll be giving you guys that same presentation today.
Yeah. Okay, I think you should start.
Okay. Imagine waking up one morning with a sharp pain in your lower back that shoots down your legs. You go from doctor to doctor, specialist to specialist. They run scans and order tests. All come back inconclusive. Months go by and your life no longer feels like your own. Brain fog and depression dull your mind. Once active, you can now barely take a few steps. Years pass and you learn to live with the pain, undiagnosed for so long that treatments are no longer effective. These symptoms are often mistaken for a variety of illnesses, but the real cause? Lyme disease. In 2023, 89,000 cases of Lyme were reported to the CDC, yet experts estimate that nearly 500,000 Americans are infected annually. Over the last decade, Lyme disease cases have more than doubled, driven by factors such as climate change and aging population. Caused by the genus Borrelia, Lyme is transmitted through the bites of infected ticks, commonly found in grassy and wooded areas. The earliest indication of Lyme disease is the distinctive bullseye rash, which only appears in around 70% of patients. Although late-stage symptoms such as facial palsy or Lyme carditis are more recognizable, these lifelong complications can no longer be treated effectively. Existing tests detect antibodies, but because the Borrelia bacteria rapidly spreads to joint and cardiac tissue, antibodies take over 3 weeks to develop, leading to a lack of successful diagnosis and frequent retesting. Introducing Lancet, a novel diagnostic platform designed to enable rapid and reliable point-of-care testing for Lyme disease, coupled with a therapeutic system designed to combat antibiotic resistance. In order to better understand the spread of the Borrelia bacteria, we contacted Dr. Nicole Baumgarth, the director of the Lyme Disease Research Institute at Johns Hopkins University. She introduced us to the upcoming use of borrelial surface proteins in vaccine development. Through further research, we found CSPZ, a lipoprotein that persists in the bloodstream long after the bacteria have moved into tissue. Our assay begins with the aptamer-based detection of the CSPZ protein, producing a specific detectable DNA sequence through proximity-dependent ligation, or PDL. To increase sensitivity, we amplify this DNA product using recombinase polymerase amplification, or RPA. The amplified DNA is then recognized by the CRISPR-Cas12a endonuclease, leveraging its collateral cleavage activity to visualize results on a lateral flow assay.
First, we utilized PDL to detect the presence of the CSPZ protein. If CSPZ is found in the patient's bloodstream, aptamers will bind to the protein, after which a bridge sequence brings them in close proximity. A ligase protein and DNA polymerase then act upon the DNA to create a double-stranded DNA product. On its own, PDL generates a limited number of ligated DNA products. Thus, to ensure the output can be detected, we amplified this DNA through RPA, an isothermal process that utilizes a recombinase enzyme and primer complex to invade the PDL product. Unlike PCR, which requires bulky equipment and can take hours, RPA achieves high sensitivity in as little as 20 minutes with minimal instrumentation. Our initial PDL experiments using literature values proved inconsistent, which our ODE model confirmed was due to low aptamer concentration, producing only about 0.6 copies per reaction. After increasing the aptamer concentration 1,000-fold, we achieved consistent, visible results in our PDL RPA gels. Using ImageJ software to analyze the integrated density of fluorescent bands, we were able to quantitatively determine assay activity to remain functional at protein concentrations for 150 days post-infection. While designing our RPA Cas12a workflow, We noticed that existing tools were often fragmented and inaccurate. To solve this, we developed Casper, an integrated tool that designs RPA primers and CRISPR-Cas12a cRNAs by ranking candidates using a composite score based on more than 10 features. Using a machine learning pipeline that combines logistic regression and permutation analysis, Casper quickly identifies the most effective designs. In testing, our Casper-generated primers showed stronger amplification and extended assay viability by over 100 days post-infection, outperforming existing design tools. We further validated our designs by testing with bovine blood, where we did not observe a significant drop in assay activity, confirming that our assay continues to remain viable up to 250 days post-infection in physiologically relevant conditions. With ample DNA produced, we needed a way to visualize detection. While RPA is highly sensitive, it can yield false positives. The CRISPR-Cas12a detector system resolves this by cleaving single-stranded DNA reporters to emit fluorescence, enabling rapid and specific detection. The Cas12a reaction generated a strong fluorescent signal comparable to the positive control, confirming successful collateral cleavage and accurate detection of the amplified PDL-RPA product. Casper's cRNA sequence also slightly outperformed our sequences designed in-house, showing that Casper's co-design of RPA primers and cRNA enhances assay activity and reliability.
We integrated PDL, RPA, and Cas12a into a single 3D printed microcentrifuge tube to optimize point-of-care use. Each reaction is separated by paraffin wax layers set to specific oil-to-wax ratios. When a finger prick of blood is added, the dehydrated reagents activate, and as each reaction completes, the next wax pair melts to trigger the following step. We conducted preliminary tests to assess one-pot feasibility. Using colored water layers at 10:7 and 10:8 oil-to-wax ratios, heated with a frugal path simulating reaction cycles, we found each layer melted at its designated temperature with no cross-contamination, confirming wax as a reliable release mechanism. To enable accessible on-site testing, Lancet uses a lateral flow assay instead of costly lab equipment. By replacing the fluorescent reporter with a fan biopsy probe, successful reactions produce a clear test line on the strip. After the Cas12a reaction occurs in the one-pot tube, the mixture is applied to the LFA, where anti-FAM antibodies capture the labeled products and generate distinct red lines, providing an immediate and easy-to-interpret visual result. On an LFA, our experimental sample showed a clear test line confirming FP-reported cleavage in FAM release, while the control displayed only the control line, verifying no cleavage without target DNA. The successful integration of our system demonstrates the specific detection of CSPZ in low-volume samples, highlighting Lancet's potential as a rapid, accurate Lyme disease diagnosis.
Even with improved diagnostics, current treatments for Lyme remain inadequate, relying on antibiotics which not only contribute to antimicrobial resistance, but also fail to address post-treatment Lyme disease syndrome, a debilitating condition where patients endure persistent fatigue, pain, and cognitive impairments. Lancet offers a novel approach through a multiplexed CRISPR interference or CRISPR-I system to silence critical genes, effectively killing the bacteria while preventing the exacerbation of resistance. Expanding on our 2024 Project SHIELD, we advanced our experimentation with CRISPR-I, which utilizes a deactivated Cas9 enzyme along with a unique guide RNA sequence to effectively downregulate specific critical genes by inhibiting the binding of RNA polymerase, preventing downstream transcription. Choosing to target the genes DETA and ARCA, we then identified the most effective sgRNAs for each gene, finding that sgRNA 60.5 and sgRNA 54.3 successfully downregulated each gene respectively. We then multiplexed our CRISPR-I system by targeting both genes simultaneously. The system exhibited strong repressive capability with an average repression of 76.01%. We then tested the CRISPR-I system in E. coli, transforming cultures with either dCas9 alone or the dCas9 sgRNA complex combined. Samples expressing both showed a 9.5-fold decrease in viable biomass and over 60% cell death, confirming CRISPR-I's potential as an effective antibiotic alternative. In the future, we plan to deliver our CRISPR-I system via lipid nanoparticles, which encapsulate and deliver cargo effectively, with a protocol we've developed specific to our system. During our preliminary research on Lyme disease, we contacted Dr. Thomas Hart, a microbiologist at Johns Hopkins who emphasized the lack of familiarity surrounding Lyme disease and tick bites, a point that was continuously highlighted through further conversations with stakeholders. Inspired by their insights, we developed over 75 educational programs, and through interactive experiences, we deepened participants' knowledge of Lyme disease and synthetic biology. Our workshops, boot camps, and outdoor events provided direct hands-on participation, while our webinars, PSAs, and presentations taught in an accessible format. Building on this educational framework, We prioritize making science and technology accessible to marginalized groups, accomplished through our exceptional science program for neurodivergent students, our Flowers for the Future synthetic biology course for Afghan women, and our cell-free system that made synbio experiments accessible for under-resourced communities. Integrating stakeholder feedback into every stage of our project allowed us to continuously learn from experts, enabling us to best empower our community to fight against Lyme disease.
By creating a rapid diagnostic, novel therapeutic, and targeted education initiative, Lancet provides a comprehensive response to the most common vector-borne disease in America. With Casper, we've created a tool for the broader scientific community to advance molecular diagnostics. By getting results in both our diagnostic and therapeutic, preliminary viability for our system as a whole is confirmed as a success. Lancet, piercing through the barriers of Lyme disease.
Should we say
Thank
you?
Thank you.
Thank you.
Thank you guys. That was great. All right. Thank you so much for sharing. So I will open the floor to questions in just one moment, but I was hoping that you might be able to give a little bit of background. I'm pretty sure most people in the BioBuilder Club have heard of iGEM by now. But do you guys want to give a little bit of background and introduce kind of the iGEM program at Lambert?
Yeah, I would love to. So Lambert iGEM is like a synthetic biology program that we offer here at like Lambert. So it's based on the iGEM competition, which is a synthetic biology competition that is hosted in Paris every year. So in this competition, we submit a wet lab project, a dry lab project, which comprises of software modeling, and we also have human practices. So we're able to combine different skills all into like one different category in a competition. And for us, this was our project over here. And at Lambert iGEM, we spent our whole year like researching this problem and like preparing it with our team. And we're actually really proud of our results over here. So we're glad to be able to be sharing it with you guys today.
Thank you. That was great. Yeah, so iGEM, for people who don't know, stands for the International Genetically Engineered Machines Competition. And it did originate at MIT from the Biological Engineering Department, where BioBuilder itself originated also. And yeah, so iGEM now has grown into a very large international competition with the high school division, run by the amazing Janet Stanovan of Frugal Science Academy at Georgia Tech. And so I was just so glad that you guys were able to share. So you are, I also was hoping you could mention, you are one of the very few high school iGEM teams in the country. And recently you were featured on 60 Minutes. So you guys have anything to share about that before I open the floor to questions?
Sure. So actually, we were really surprised because we actually didn't know about 60 Minutes until Mashira told us that it's been in the books like for a while, like they've been planning it. And we actually couldn't tell anyone about it until like the press release day. Like we could tell like family members, but it wasn't like we really didn't know whether we were gonna air it, like it was gonna air or not until they like specifically told us. But I just think the experience was so cool, like getting to see what everyone thinks about our project and thinks that we could really make an impact. So I think that was really cool seeing how everyone reacted to the thing.
It was awesome to see you all on TV. All right. So any students, teachers on the call who have questions for the Lambert iGEM team, and we'll be able to see you better if you stop the screen sharing.
Okay.
You're back. Okay. Anyone have questions?
If no one else, if no one else, sorry, because you got to mute. Okay, I'm so sorry. If no one else has a question, I know that Veda did BioBuilder, and I wonder if you'd like to hear a little bit about her experience with BioBuilder.
Yeah, and then I have
like
questions
gave her
if
some
no one
of the
else.
experience she needed to be successful, and I'll mute.
Yes. Hi, yes, I did do BioBuilder. So essentially how our application process works here at Lambert is we all submit like a project proposal, right? And so on that proposal, once you write it, we have to fill out like this little Google form. And on there, it's like, do you want to be part of the iGEM seminar team, the competition team, or both? Like, which ones would you like to try out for? And so when I first tried out, I tried out for both, and I was put on the seminar team my freshman year. And essentially, the seminar just does biobuilder stuff. So I was kind of just introduced to the basics of synthetic biology and what it was like to even create a project based on biotechnology. And so after, like, I think I created something about like some gut microbiome thing. I don't really know. It's like 2 years ago. But essentially, what BioBuilder did for me is it created this kind of environment where I was used to like making ideas and understanding the basics. So then when I reapplied my sophomore year, I actually made it onto the competition team because I had that background knowledge from BioBuilder. So I think it's a really good program for people that are just starting to get into biotech and not really knowing how to jump into iGEM. So
Yeah, I was also part of the BioBuilder program my first year, and I think my experience was really similar. Like BioBuilder for me was my first experience with anything synthetic biology related at all. So I think that was just really informational. It was a great starting point for getting where I am now. And it was also my first time working with a high functioning team, which I feel like really helped getting into competition team last year and this year too.
That's awesome. Yes. Anyone else here have questions?
Can I ask something?
Yeah, of course.
Yeah.
Yep.
So did you have all of the resources you used in your projects at your school, or did you have to order anything from anywhere?
So for all of our wet lab reactions, we had to order pretty much everything because we started from scratch for this project. So most of other than the CRISPR-I system, for all of our diagnostic, we had to order everything. But we got a bunch of this through fundraising. Like we held a summer camp that helped us fundraise to buy our reagents and help us go to Paris. But yeah, we did have to order quite a bit of stuff.
I will also interject here just so that everyone in the BioBuilder Club has a little bit of context. So Lambert has had an iGEM team for a very long time and already had the actual like equipment and lab space. And so when they talk about fundraising and buying materials, those are a lot of, reagents that are used during experiments and consumables that will have to be replaced each year. And so there is a lot higher a cost in participating in iGEM, especially the startup cost. And I don't know if you want to add to that, Ms. Scherer.
Yeah.
We turn the camera to you.
Yeah.
Oh, I do, yeah.
Hello, how are you? So yes, we were very fortunate that Janet had already established a lot of our equipment, and we are very fortunate to be in Georgia, where Georgia Bio is a resource where labs can dump old equipment they don't need anymore. And so we've had donated equipment, we've had equipment that's been purchased through the county through Perkins money. Being part of our career tech program, we get money for equipment, and some of our equipment that we use for biotech works for that. And then our summer camp, we had 60 middle schoolers. We're in an area where I think partly because of iGEM and biotech, it's synthetic biology is very exciting to even the middle schoolers. So we filled up a camp and raised money that way for our reagents. And then the students generally pay their travel costs.
Thank you for sharing that. I hope that gives some context to teams.
I
And
always say iGEM is expensive to buy a builder. Like I think for sure we're fortunate to live in an area that's, we're able to do that. But I think you can design a project, you learn everything you need to know about synthetic biology. And then if you have the resources, you can execute it, but you don't have to. You can just think through the design and do the poster presentation and be part of that community without having to actually buy the expensive reagents. And that's the beauty to me of what you do. And sorry, last thing, the teachers who say, well, I don't know anything about synthetic biology, you don't have to because you've got the mentors. And I think that's the beauty of your system.
yes, that was a good question. Thank you. Yeah, so with the BioBuilder Club, like Ms. Scherer was sharing, one feature is that you can really get exposed to synthetic biology and learn that whole biodesign aspect, which isn't usually in the high school curriculum. And then many of our BioBuilder Club teams do come back year after year, and some of them in their return, if they choose to work on the same project, then they already have the design, and then they'll have time to work with a mentor to actually build and maybe test that project. And so there are certain kind of lab equipment resources that are pretty standard that you need for doing synthetic biology, basic molecular biology equipment. So the BioBuilder Club handbook does kind of detail that. You can always reach out to me if you have questions about that. Anyone else on the call have any questions?
I have one.
Anna.
Hey, so there's 20 of you guys in the team right now?
Yes.
So how many are returning for next year?
So we have 9
Nine.
members who are returning for the upcoming year, and we selected 11 members actually. Our first main meeting was actually today, and it was a big success. So it's great to see the new members coming to our team for upcoming.
Okay, are you guys doing a brand new project next year? Or are you going to continue this?
Yeah, we're thinking of making a brand new project. We're currently in the process of researching and seeing what are the next steps needed to create a great project.
Okay, that sounds great. Well, congratulations, everyone.
You.
I guess I'll jump in with my question, building off of that. So with the BioBuilder Club, I hear from a lot of students and teachers and mentors, as students are kind of exploring the decision-making process, and as it's just brought up there, I would love to hear, and I know lots of people would love to hear, how do you guys make decisions? So how do you decide whether you're going to continue a previous project, start a new one? I remember from the 60 Minutes segment and watching your presentation right now, it's a very high risk project. So what factors go into that decision? Do you have a formal decision making process? You know, it's 20 of you on the team. How does that work? Tell us.
Okay, so basically, how we kind of come to an agreement about the idea of a project is we separate ourselves into groups like weekly. And what we do is we do research and we come up with like potential ideas or like little proposals and then present them like weekly. And so what happens after that, we go through and like we talk about it and we're like, okay, which projects do you think we should continue on for next week, or which ones do you think that we don't like and we'll like eliminate them? And so we just keep doing this until we develop like a more complex project, and we're kind of this way, like it's almost like everyone's integrated in the idea of like to create the idea, and everyone's on the same page about everything rather than like it's one person coming up with an idea and people agreeing with it, if that makes sense.
Yeah, ultimately all our decisions, they involve the entire team. We all sit in a circle and we discuss and we come up with something that everyone agrees with.
Sometimes it comes down to a vote.
Yeah, it does.
Yeah.
No.
The whole
Vote.
ideation process overall took us about like 3 months last year. So it's not like a short process at all. Like we're constantly doing research and creating new ideas and like starting from scratch. So it's kind of like a tedious process, but I think what comes out of it is definitely worth it.
And how with that decision making process, with you know the team voting and anything, how do you involve consulting your scientific advisors and incorporating their feedback and opinions versus the weight of the team's direction?
So I take to y'all cover. I try very hard to let it be their direction because they really are way more creative. They're way more risk takers than I would be, and I think that's the beauty of this kind of work is that the students get to direct it. When else in the world? And you know, Chloe, from being in the real world, you never get to pick your own project. You're in someone's lab and they give you an assignment, or maybe you can develop something within the structure of it. But this is like the only time ever that they'll be in a sandbox. So as long as it's safe and it's relatively feasible, like I'm game to try. My gears are turning thinking about a proof of concept for each step. So just if this doesn't work, what can I do to help them get to the next step? If that doesn't work, what can I do? And I'm actively trying to do that as we go through the course of the project.
Also, if I could add
I think
some,
that's really great advice.
sorry. I think when you talked about the researchers, like I think we definitely talked to more researchers and like stakeholders further into the project rather when we're trying to like actually create the project. So like maybe in like the last couple of steps of like finalizing an idea, we would talk to a researcher, but in the beginning steps, we usually don't.
But I would like to say the researcher in this case that we spoke to said, this is a terrible, no, he didn't say terrible. He's like, this is not, like I said, so you're telling me you're going to do 4 new assays that you've never tried before and you don't know anybody even to ask about them if they don't work. We're like, yeah, that's probably not the way you should do science. And that isn't the way you would do it if you were, you know, actively in a university. But that is, again, the beauty of high school. If you're willing to take the risks, it's really fun.
I agree that that's a really fun aspect of iGEM and the BioBuilder Club because you're right. In the real world, especially if you're entering research through conventional paths in terms of joining someone's lab, that's not an option. I actually started with iGEM as an undergrad before now working for BioBuilder. So it's a nice little fun circle there. And it really is amazing to be able to introduce students to the creative side of science so early in your journeys. So anyone else have any other questions? I don't want to monopolize the Q&A here.
Can I ask another question?
Yeah.
So obviously, like for both iGEM and even for BioBuilder, these projects require a lot of researching and a lot of information, like a lot of knowledge. So how do you get your knowledge? Like, how do you understand how these biological processes work? Like, where do you research? How do you find the information?
So I think a lot of our knowledge comes from our older members on the team. So initially they introduced us to these concepts of synthetic biology. And as we learned, we were able to be exposed to like new processes, new things that we research on our own. So a lot of it comes from our older mentors. And additionally, all of us on the team, we're very curious. So like as we learn more things, we search about it, we look into research papers, we look into different processes, different iGEM teams, and we're able to expand our knowledge base on this topic overall.
Yeah, I feel like
Yeah, okay.
I feel like even if you come into iGEM or BioBuilder not knowing much, that doesn't really matter as long as you're willing to keep learning it. Just like keep looking into papers, see what you're interested in, and then keep building on that. And that's what matters more than whatever foundational knowledge you have.
I think another big factor is that we all spent a lot of time researching, just in general. Like because we spent so much time, we've kind of like succumbed to research and like understanding them a little bit better. Like we have 2 hours, like a class period, 2 hours in our school schedule just dedicated to research for the first 3 or so months. So it's like it seems like a lot, and it is a lot, but it really helps us like understand what we're actually going into.
That's a great answer. I would say also for the BioBuilder Club, everyone does have a copy of the BioBuilder textbook, which in addition to going through synthetic biology and biodesign, does have a chapter on basically the tools of molecular biology. And the cool thing about synthetic biology, and in the BioBuilder Club is that we teach this top-down approach. So this abstraction hierarchy, starting at the highest level of the system. So what do you want to do? And then this really allows you to build down from there rather than starting the nitty-gritty details of assays and biological processes that you might not know about yet.
Okay. Or if we can move.
Did you want to add to that, Ms. Sher? You just
Yeah, well, and I don't know if this is pertinent for BioBuilder. Even in that research process, there's been times where maybe they didn't use that piece of information then, but a couple months later, somebody will say, "Oh, well, I was looking into an RPA reaction, and we didn't use it for this, but we can use it here." So that time is never wasted. The reading and learning and absorbing, you might use it in different ways.
I had another question.
Yeah.
So, the first one is, what do you guys do if one of your team members isn't researching and they have paid their dues and everything?
That's mostly Miss Sher in the captain's jurisdiction. Like I myself wouldn't do anything,
Uh-huh.
but it's Miss Sher. Do you want to talk
Sure, no
about
problem.
that?
I joke that I'm just like the HR person for the team because that's exactly what I handle. And I would say the vast majority of the issues that come up are things like this. There's never been a group project where everybody contributed equally. That just like doesn't exist. But where the way we handle it is if every project has a committee lead. So that committee lead is in charge of the assignments. So if that person isn't completing their assignments, the committee lead will cue in the captain. The captain will cue in me. If it doesn't get better, I will have a one-on-one conversation. So the first time would be like just a verbal warning. The second time, I actually found a performance improvement plan, just like you'd have at a company. And we complete a performance improvement plan together, which is a little bit scary. And I just sort of hold on to it. And by the third time, and I have not had to do this, I think that they have in the past, then that's a parent call. The parent comes in, we talk about like, if these things improve, then the student may stay on the team. These things don't improve, they may not stay in the team, and there are no refunds. That's just how it is. It would be the same if you were in a job. When you're working on a team like this and you're working at this high level, you've made a commitment to each other. So sorry,
Yeah.
it's not an uncommon problem in groups.
Okay, thank you. And I had one more question. So you said that you used RPA, right? Yeah, how did you guys RPA is known for like false positives from primer dimers. I'm sorry, primer dimmers, right? So how did you reduce the formation before Cas12a?
Is Sean on the call? Sean,
I think we could talk
can,
about
could you
Cas-
Yeah. Or actually, Veda, you can go ahead.
or
sorry, but go ahead.
essentially what we did, like we have this software tool called Casper, and what it does is it designs the most optimal RPA primers and Cas12a crRNA. So when we started doing experimentation with the Casper generated primers, we saw that there was a lot more specificity within RPA and like it was a lot more successful, and we didn't see as much formation of the secondary structures that you were talking about.
Okay, thank you.
Does anyone have any questions?
Questions, anyone?
I'll ask one more question.
Okay.
So sorry, I think this will be helpful. So it sounds like you have a very democratized decision-making process, but that it's also really beneficial to have some sort of leadership structure. Could you say more on that?
Yeah, so our team is comprised of two captains. In our past team, we had two captains, but currently we have three captains in our team, and each of our committees are set up into committee leads. So the way our team works is our captains and advisors set out clear guidelines on what needs to be done and how our structuring all works out. And our committee leads make sure that each person who's on a committee is on track on what assignments they're doing. And we set like clear deadlines onto when we expect certain tasks to be done and how we expect our meetings and progress to be moving on. And I think definitely since our planning was for a whole year, that definitely helped us with pacing forward as we went through our different projects and as we went through coming up with our results and our experimentation. So structuring like that definitely helped.
We also on the team have a bit of like a system just to help like new members. It's called like Bigs and Littles. It sounds a little bit dumb, but it's basically when a new member joins the team, they get paired with like an older, more experienced member so that they can like help guide them through the program, like help them understand like what's expected of them and how they should be acting on the team. So I think that really helps actually because just like having someone you can easily talk to and having someone who like you always have access to ask questions to and like learn more from is definitely like a useful resource.
I definitely think it's really important, especially as we go into the later, like the fall season, because as we're like onboarding the newer people, like iGEM isn't as big as a commitment as it is in the fall because it's getting closer to Jamboree. Like we spent so many hours and like days working on things that we need to get finished. So like I feel like having these bigs and littles and having this whole hierarchy in place really allows for them to understand what they're really getting into.
So it sounds like near peer mentorship is really important in onboarding new members to your team. That's awesome. That is solid advice. So most of our BioBuilder Club teams right now are, if they haven't started working with a mentor yet, still coming to a decision on kind of what topic. And I know with the literature research presentation, we talked about how to kind of conduct primary literature research, but what sort of techniques and resources do you guys use when you're in that early process and still brainstorming and trying to come up with ideas, like you mentioned with the project proposals?
So definitely research papers are probably our number one go-to whenever we want to learn something new. And definitely like while we're researching into a topic, looking into past projects for iGEM specifically, looking to past projects helps us grasp an idea of what to look forward to. And relating to BioBuilder and things like that, looking up like the definitions and coursework that are related to the concepts definitely help when setting up what's needed to be done for a project.
Kind of expanding on what Shashwat said, like especially like the projects that either like won a best award for something that you want to go for. Like I remember last year when we were trying to like do our modeling and stuff, we would look at other modeling things to see what other people were doing and see how we could possibly do something similar or something like relevant, so we know like what quality we need to get what we wanted to achieve.
I also think that a lot of the research process in the beginning needs to just be driven by like what you're interested in. So like if you find like a specific reaction or like a specific disease that really, really interests you, like looking further into that and looking what you can do with that specific little thing helps. Because if you're just looking for like a broad little thing, like research topic, that you're really not going to find anything too quickly. So I think just like looking for something specific at the start kind of helps.
Yeah, to build off that, I think before you come up with a solution, you really need to find the problem for that first. And for that, looking at news articles really helps, maybe looking into friends or family members who have some sort of rare illness or something, and then basing your project ideas off of that will help in the future, like determining what you want to work on.
And I would mention reaching out to stakeholders. You
Yeah.
could cover that. So like at some point they reach out to researchers in the field or people that are relevant to that field too for thoughts.
After we have an initial basis of research done is when we kind of start reaching out to researchers to see how feasible is our project idea. Do you think it will have a real world impact? And then the further we go into that, like for our Lyme disease project, we did end up reaching out to Lyme disease patients who told us how maybe educating them or educating healthcare professionals about Lyme disease would have helped with their diagnosis and treatment process. You want to add something?
Also no, but also with stakeholder feedback, like one of the main things that like can help you in your project is just emailing as many people as possible, as humanly possible. Because obviously as high schoolers, we don't have the largest background in synthetic biology, and we're not going to be able to like automatically understand everything. So just anyone related to your field, anyone who might have a specialization, just don't be afraid to reach out to them and email them, because a lot of the times they're busy, they might not respond to you. So just a lot of like broadly approach anyone you can who you think might be able to help you.
The worst thing that can happen is you won't get a reply.
I think maybe like every 20 emails we send, we get one response.
And I will say, and just something I've noticed is keep the email short. They're very busy, and keep the ask small. Make sure that you're not asking them to explain something to you. I try to encourage people to say, this is how I understood your paper. Am I correct? So it becomes just a little bit easier for them to respond to you.
And if they do email back and they're willing to help us, then we set up calls with them to maybe discuss our project further.
So for your Lyme disease project specifically, I don't know if you can, because you mentioned looking at kind of different diseases and things. So what made you go specifically in the Lyme disease direction?
Yeah, so that's kind of an interesting topic. So we started actually during our like 3-month period at the beginning while we were doing research, we figured out kind of like a wet lab like workflow, like a few different reactions that we found like could function very well. And we found that it was good for like diagnosing like illnesses that leave behind proteins. So initially, I think we were looking into sepsis because that was like a very relevant issue. But when we did further research into that, we found that that had already been like a similar project had been done in previous years. So then we were just looking for highly relevant issues that like our diagnostic like workflow could be used for, and we found Lyme disease.
I think we were originally like actually looking into tick-borne diseases first, trying to find a way to combat all tick-borne diseases, but then we like narrowed down to Lyme disease too.
Any final questions from everyone else? All right. Well, thank you very much, Lambert iGEM team. Have any closing advice for students?
Advice?
One thing I would say is just like, don't be scared. Like, I feel like the first proposal I ever made was really like, it didn't really make a lot of sense, but like, that's what even got me on this journey in the first place. So like, just make sure that you don't like, just be ready to like immerse yourself in biotechnology if what you're getting into.
And don't feel like unqualified to be doing this, because I don't think anyone when they start out is actually like good at it, understands it very well. Like, don't be afraid to approach it just because you don't know anything about it.
I agree. Like
And-
in the beginning of this year, like all like I don't know about Joshua because he was on the team last year, but like all of us, we were all newbies. We didn't know anything. Like the learning curve was so big. We learned so much. Like right now, like I feel like we just feel like so much more accomplished after going to Paris and doing all this that we have done. So
And kind of to build off of that, just push yourself to keep learning. Like make sure you're always taking in new information and kind of thinking about how maybe you could apply this in the future.
Yeah, I agree with that. Curiosity is probably like the best advice we can give you guys because a lot of us we learned so much over like the past couple of years in this program and immersing yourself in like research papers and like different topics with like-minded people on a team is definitely like the best part of being part of this experience.
Advice, Arjun?
I mean, honestly, everyone said everything for me. But
Okay.
yeah, my biggest thing is that like when I walked into my first IGEM meeting last year, I was definitely pretty scared. I didn't really know anyone. I thought everyone else was smarter than me. And then I slowly realized that, all the other newbies are in the same boat and we're all learning everything together. So never be afraid to contribute and never be afraid to share all your ideas because every idea matters.
it's pretty.
And feel free to reach out to us at any time. You can
Yeah.
find, yeah,
We'd
for
love to
sure,
help.
for sure.
If you
Just
go on our
email
wiki,
me.
you can find our email.
Yeah. We have a team email. We're happy. It might take us a minute to see it, but we're happy to respond if we can be helpful.
Awesome. Well, thank you so much. And congratulations on your success in Paris. And thank you for sharing, everybody.
Thank you.
Thank you, Chloe, for everything you're doing for high schoolers. Without BioBuilder, I mean, so how many BioBuilder teams are there nationally?
Right now, we have 81 teams.
Crazy. That's
That's
huge.
really good.
That's amazing. Yeah.
It's a lot of students.
A lot of students, but that's the difference. That you're making a real difference here in the US for sure. Thank you.
Well, thank you. Thank you, everyone. Have a good night.
Thank you. Have a good night.
And come on Thursday to hear Anna White's presentation on plasmid design. Bye, everyone.